Thank you. Good afternoon. I want to thank the organizers for the opportunity to present again this year. We are Sentien Biotechnologies, a cell therapy company based out of Lexington, Massachusetts, clinical stage, venture-backed. And I'm excited this year that we'll be able to give an update for those of you that might have heard of us in the past, show some clinical data, an overview of our clinical data for the first time, which we're excited to present. And for those of you that are new to Sentien, we'll tell you a bit about who we are. So we are a cell therapy company using mesenchymal stromal cells. We do it a little bit differently than how people have done it in the past. Instead of injecting the MSCs IV or into the tissue bed directly, we actually encapsulate them into an extracorporeal device. So it's really an ex vivo cell therapy, and I'll talk more about why we do it that way. The clinical data that we'll be showing is related to our first-in-human study for dialysis requiring acute kidney injury. The data generally supports our overall therapeutic hypothesis, which of course we're very excited about, showing an anti-inflammatory and immunomodulatory effects. Hallmark signs of MSC biology is what we'll be showing. So we're looking at AKI as our first indication, but we really think broadly about the critical care space as a starting point. So other indications also where single factor agents are unlikely to be effective based on the complexity of disease. Things like the inflammatory surge from burns or ALF, ARDS, or trauma, those types of indications where single-factor agents are not going to be effective. So we are in the process of raising a $30 million Series B to advance the program, and I'll give you a pipeline slide towards the end. So many of you are familiar with MSCs. The real benefit behind our therapeutic hypothesis with MSCs, they're very broad acting, so they secrete a lot of different molecules that collectively lead through multiple different pathways into a regenerative type of tissue healing response. And it's really that nature of MSCs which has brought so much attention to them over the years. In addition, as a living tissue, they are dynamically responsive. So when they're given cues of pro-inflammatory signals, they will change their secretion profile dynamically, not quite in real time, but within a therapeutically relevant time frame to adapt to the signals they're getting. And so it's really those two features that have given people a lot of interest in MSCs over the years. The challenge has been when you administer them, they tend to have a relatively short half-life. So the therapeutic exposure, the area under the curve of these secreted factors is generally relatively short. And so our technology really set out to try and solve that problem. And how we do that is, as I referred to earlier, putting the MSCs into this hollow fiber device where the MSCs are on the outside and blood flows through the inside, allows for the cells to remain viable. You get that communication between the blood compartment and the cells so they can dynamically respond, and they're viable for long periods of time. We can put many, many millions and millions of cells into these devices and really get that sustained duration of exposure for the secreted factors like I was describing. As one benefit, I don't think I have a pointer here, but oh yes I do. As one benefit with the blue caps there, that's actually a sampling port, so we can sample from the device during treatment and really begin to characterize the biology of the MSCs as they're interacting with the blood. It gives us levels of insight in the pharmacology and the biology that really can't be done in any kind of in vitro setting. So it's really those factors that have led us into the clinic. The first indication is acute kidney injury, as I referred to earlier. We view ourselves as a standard add-on to these clinical types of modalities. So where you have a blood circuit in clinical use, think dialysis, think extracorporeal membrane oxygenation for lung injury, photodynamic therapy, plasmapheresis. Anytime there's a blood line that's being taken out of the body for some kind of treatment and then being delivered back in, we can take our product and just add it right back into that blood line. So it's really a standard add-on to existing standard of care therapies. Overall, that represents about 1 million patients per year in the U.S. that get some kind of treatment related to extracorporeal modalities, so it's a relatively large opportunity. Our initial indication is dialysis requiring acute kidney injury. These patients are very, very sick. What they get today is called continuous renal replacement therapy, or CRRT. It is 24/7 dialysis. Their kidneys have effectively shut down, and their blood needs to be filtered out from the toxins that would otherwise be cleared by the kidneys. However, this provides absolutely nothing towards the underlying inflammation that these patients are experiencing. So it's really this need for addressing the inflammation at its root, which provides the clinical opportunity for a product like ours in this patient population. And with AKID, it's a really, really expensive. In fact, it's the most expensive diagnosis in the ICU setting. CRRT is a very labor-intensive activity. So our goals, in addition to reducing the mortality rates, trying to help with morbidity rates as well, is really trying to shorten the amount of time these people are on CRRT by promoting this wound healing I was describing earlier. So those are our clinical goals. As we go into the trial, we're measuring, obviously, a number of different types of things, including some of these pharmacoeconomic factors as we go on through development that we can be using that with payers and hospital systems. So I have a little bit of data at the overview level. The phase 1b data, what we're showing here are just representative cytokines that we've selected because they are hallmark cytokines with respect to MSC biology. So TNF-alpha and IL-10, we call them the usual suspects in the MSC field. So what we're showing is in the blue is our therapy. And so the way we think about this from a dosing perspective is both how many cells we have in our bioreactor, but also how long that bioreactor is integrated into the blood circuit. Because at the beginning of treatment, we integrate our product, and in this case, in this study, after 24 hours, then we take the product away. So the blue shading here is showing our low dose for 24 hours of exposure. Once the product is taken away, then we're still following the patient, measuring peripheral blood over the following days. And so what you see with things like TNF-alpha and IL-10 are very encouraging that some of these effects begin during treatment, but they persist even out to 7 days. CRP is another acute phase protein common in the inflammatory space. So collectively, what we're showing here is what we like to think of as blood conditioning, but also a reprogramming of the immune system during the 24 hours of perfusion with effects that are continuing on well beyond the product therapy itself. So generally going from a pro-inflammatory markers are going down, anti-inflammatory markers are going up. And again, we're giving you just a representative example here. There's many, many cytokines that we've been evaluating. So it's really not a cytokine story. It's really more of a systems biology story where we're modulating the broader immune system and the multitude of inflammatory pathways that are being affected by this disease. So it's really hard to appreciate on this slide, but what we're showing here is a broad list of inflammatory pathways. So we took the entire cytokine data set that we had from day 3 and subtracted that out from baseline. And basically what's being shown at the pathways level is broad spectrum inhibition of these inflammatory pathways with our product, as shown by the blue in these columns. The orange is showing the same pathways that are active or remain active in the sham group. You'll notice there's one star that we have here. There's a patient in the sham that looks a lot more like treatment. That was really almost not by design, almost a nice control for us because that patient had sepsis that was resolving at the time of treatment, at the time she got the sham product. So it's almost like a roadmap for what getting better looks like, and so we were encouraged to see that the blue line from that sham patient who did get better looked a lot like our patient population as well. So the reason I like to show this, again, cytokines are great. It gives you a sense of what's happening at the molecular level. This is really mechanistically the broad view that we think is important for why a cell therapy like this is needed for these complex diseases. It's this broad inhibition of multiple inflammatory pathways. As we think about translating this specifically for the patient population, what's shown here in this cartoon was from a nephrology paper, a review article, showing the various players at cytokine and cellular levels that are involved in both early injury, late injury, and in the regenerative healing response. So as we looked at our product, active versus sham, what we're noticing is that a lot of these injury markers are going down relative to controls, and the healing markers are going up. So obviously it was very encouraging for us to see this on the broad spectrum. Really what we like to say, and our scientific co-founder has been saying this for about 10 years, MSCs promote wound healing, and that's kind of what we're trying to show here, is this acceleration from an injury phase into a wound healing phase with this product. So it really is, again, an illustration of this broad level of immunomodulation that's taking place. So just by way of example, you have Th1 going to Th2. You have some upregulation of regulatory T cells, again, with the cytokines that are commonly assessed in this type of setting. So from a pipeline perspective, we are a small, early-stage biotech company. So we've been focusing on AKI as our lead indication. Where we'd like this to go is really thinking more about the critical care space as our next wave of development. So the company pipeline focuses on the lead program and taking that into a dose escalation and then moving it into a proof of concept study in a phase 2b, but also starting other programs also in the critical care space where you have sister indications like burns or trauma or ARDS, where they also have this massive systemic inflammation that could be treated by a product like ours. So we'd like to get into additional programs in the ICU. We'd like to be able to show that we have a similar biological response outside of AKI in something like burns, for example, where if you have that common biological response across these systemic indications, you start to get the picture this could be used across the broader ICU space. In addition, we think about inflammatory diseases where, in the chronic setting, where this could be useful. A natural starting point would be chronic kidney disease, but we also think about the autoimmune space, where again, tamping down broad immunomodulation with a product like this could be potentially beneficial. So the team, yours truly, is humbled to be the leader of this group. We have Chris Giamini, our VP of Operations, also here with us. A lot of people here at this conference know Chris. Andrew Blair is our Chief Medical Officer. Biju Parakadhan is our scientific co-founder. He's considered one of the leading experts in the MSC field and has been for a while. It was his research that really began to move people towards thinking away from direct transplantation and into more of the secretion profile of MSCs. And then Dr. Rita Barcia on the R&D side, also an MSC expert that's been with us now for a few years. So in summary, we're very excited by what we've seen so far. SBI 101, our lead program, is beginning to show in the clinical data what we've seen also in our preclinical data, this real translation and realization of what MSC biology can do in the clinical setting if administered in ways that we think are appropriate for this patient population. So we're seeing this anti-inflammatory and immunomodulating effects that we were anticipating seeing, and we're very excited by this. Next steps are to move to the higher dose and get more patients, as well as extend into other applications as well. So the Series B of financing will be primarily driven towards getting AKI towards proof of concept, as well as extending and demonstrating the breadth of our platform across the ICU and then into chronic indications as well. So we feel like we're in a good position. We're happy to have an opportunity to talk to anyone who would be interested in either partnering with us or participating in the Series B, if that's your flavor. And again, thank you for your time and your attention.